So welcome, folks. Thank you all for being here. Thanks for knocking out that warm-up really quickly. Today we will take our lesson to review for Unit 2. The exam will be on Monday. Today is January 29, 2021, and it is the 5th day of our second unit. We've got two objectives today, as well as an important essential question that we will come back to. So I want you all to take some time to write it down so you can just glance at it and remind yourselves as we talk of what our goal is today. But the first objective is for biologists to be able to solidify their knowledge of the structure and function of cells in preparation for Monday's Unit 2 exam, and also to clarify any and all misconceptions they may have about how cells complete the Stirringer processes. So today is going to be a really engaging lesson, and I want as many voices represented as possible, so everyone should be willing to take themselves off of mute, or at the very least willing to type into the chat. And also we should hope that if anyone has any misconceptions or any confusion, that we can get those cleared up. I want you all to feel free asking questions, whether it be now in class or even afterwards. If you want to send me an email or a remind message, that will be fine as well. The essential question that I want you all to be very aware of and conscious of today is, what are the features of different types of cells that make life as we know it possible? The reason I want you to pay special attention to the essential question today is that we will come back to it at the end of the lesson, and I want you all to be able to answer that question, to give me some examples. All right, so keep that in mind. I'm leaving this up for a few more seconds so that folks have an opportunity to write things down. Oops. Okay, sorry about that. All right, so there are three, before we jump into the actual review, I want to provide some reminders about our test. So again, the test will be on Monday, February 1st, this coming Monday. Per usual, the test is worth 100 formal points. So in this class, we don't have many formal assignments outside of the tests. So the tests play a large role in your overall grade in the class. All of our tests are cumulative. And so what this means is that the tests get a little bit longer as we go because we're adding more material. So you will still have material from Unit 1 on the Unit 2 exam. The reason we do this is to better prepare you for the EOC exam in May. The EOC exam is cumulative, and it does require that you all have a good amount of test-taking stamina because it is going to be 60 questions long. So keep that in mind. We're going to keep building up in length. The tests will get longer. That way, by the time you actually sit down for your EOC exam in May, it won't feel any longer than the previous 2 or 3 tests. The best method of preparation for Unit 2 exam on Monday is to complete all of your asynchronous assignments. We aren't adding any more asynchronous assignments. There is not one for today. If you looked at Canvas, you should have noticed that. There is not one for Monday. So what you have now is what you need to have done by Monday at 11:59 p.m. But hopefully you get it done over the weekend, because the best preparation for the test is truly those asynchronous assignments, including your warm-ups and exit tickets as well, which has some good test questions incorporated in them. And lastly, our Unit 1 test was taken with a little bit of leniency. I gave you all the option of using your notes, and I also allowed some people some extended time, which meant that they, came back and finished it after they started it. We're not doing that for the Unit 2 test, and we're not doing that going forward. We've got to prepare you all for the reality of the EOC exam. Of course, you aren't able to use your notes on the EOC exam, and you are not able to blow past the allotted time. So the time that you're given is the time that you're given. It will be more than 60 minutes because the test is longer. It'll be about 30 or 35 questions, but you won't be able to say, hey, Mr. Red, I started it, but I didn't finish it in the allotted time, but the timer ran out. Can I go back and finish it? The answer will be no, because you know that once you sit down and you start, you need to finish it. Unless you are able to provide a valid excuse beforehand. But the key word there is beforehand. All right, so it can't be it can't be moments before we're starting the test and you're telling me, oh, actually I have a doctor's appointment that starts in 30 minutes, so I won't be able to take it the whole time. You need to let me know beforehand when things like that come up. And you guys have been doing a good job of that. So I appreciate you all. All right, so in this second unit, we had an additional 3 standards. So unit 1 was 3 standards. Unit 2 is 3 standards. We're going to continue to introduce more and more standards until we hit 30. Right? There are 30 standards that are covered in biology. Some units are longer than others. We will have a 5 standard unit, and we'll also have a 1 standard unit. But on average, it does average out to about 3 per unit, right? So just keep that in mind and remember to be flexible as we go. This first standard from Unit 2 was standard 1.1.1 about organelles, and it states that students should summarize the structure and function of organelles in eukaryotic cells, including the nucleus, plasma membrane, cell wall, mitochondria, vacuoles, chloroplasts, and ribosomes, and ways that these organelles interact with each other to perform the function of the cell. So this was not something, okay, thanks for letting me know, Sanswan. This was not something that we talked too much about when we first introduced organelles, but this is something that we do need to pay special attention to. It just kind of dawned on me that I didn't explain the link that I sent to the chat, but I'll send it again. So this link is a link to our YouTube playlist where I place all of our recorded class sessions. So in the case of what Santuan just experienced, he said he has an appointment at 1 o'clock. No big deal. Attend your appointment. But you also should know that we've got the lesson available to you to watch later on YouTube. All right. So don't miss those opportunities to get the lesson, even if you're not able to be present for it. Let me make sure my attendance is all good. Ashley is here. Sway is here. Cellular Adaptations is here, right? Yeah. Sully, Nico, Tiaja, and Abina. All right. So the first part is to be able to summarize the structure and function of organelles. So let's talk about that. Here we've got 2 cells. Does anybody know what the difference is between these 2 cells? There's a difference in the type.
Prokaryotic and eukaryotic.
Actually, these are both eukaryotic cells.
So one is a plant
Oh,
cell
right,
and
because
the
it
other one is
doesn't have the
Yes, I think I heard Abigail say it. One is a plant cell. I don't know if it was Abigail or Carolyn, but sorry. Plant cell on the left, animal cell on the right. Now, there are some differences, obviously, between these 2 cell types, but there are also a lot of similarities. The first similarity, what's that big purple thing in the middle called?
Nucleus.
Thank you, Janelle and Josue. So they both have nuclei. That's a similarity. What about these oranger structures that have folds in them? What are they called? I see several of them.
mitochondria.
Excellent. Thank you, Abigail. Those are mitochondria. And we'll talk about the function of these organelles on the next slide. What about those light blue structures? There are several of them in the animal cell, and I see a big one in the plant cell.
Vacuole.
Wonderful. Thank you, Melissa and Carolyn. Those are vacuoles. A couple other similarities. You can't really tell, and my arrows aren't pointing directly at them, so I apologize, but these small dots that are kind of studding this structure near the nucleus? What are those small dots called?
Ribosomes.
Wonderful. Those are ribosomes. So they both plant cells and animal cells and all living cells have ribosomes. Okay, what about the pink structure on the outside of the cell?
Cell membrane.
Yes, that is the cell membrane. Otherwise known as the plasma membrane. All right, now there are a couple of structures that only the plant cell has. The first one that we can identify is this neon green thing on the outside. What's that called?
Cell wall.
Thank you, Dijon. Thank you, Abigail. That's the cell wall. And then there's one other in the top. It's also a green, neon green. What's that called? Only plant cells have it.
Chloroplast.
Wonderful. Thank you, Janelle and Carolyn. Chloroplasts. All right, so those bottom two are only present in plant cells. We've got to be careful with that. But we can see that the others have some similarities. Now, the nucleus, the mitochondrion, the vacuole, those are considered membrane-bound organelles. That's going to be important in a later discussion. The chloroplast is also a membrane-bound organelle, so keep that in mind. But as we saw in the standard, it's not just important that we can identify the structure. We also need to know what the function of those structures, functions of those structures are. So the nucleus, what's the function of the nucleus?
The power of the cell.
What's that, Lisa?
I said on the power of the whole cell, like the mind, something like that.
Okay, yeah, now we want to be careful. I like the second phrase that you used. You said the mind, but that first phrase, power, we wouldn't typically associate power with nucleus, but the mind is a good way to put it. And Abigail adds, it has the DNA, wonderful. So in general, the nucleus is the mind. It's the control center of the cell. And the mechanism that it uses to control the cell is DNA. So good tag team there. Ribosomes, what do they do? I know this is Josue's favorite one.
Create protein.
Yes, excellent. They synthesize or create or produce proteins or make, all these different ways to say the same thing. Ribosomes produce proteins. What about mitochondria?
Creates energy.
Wonderful. Mitochondria are responsible for creating energy. They are the powerhouse of the cell. So, Lisa, this is why I wanted to be careful, because typically that word power, we would more closely associate that with mitochondria versus nuclei. So we want to be careful with that. I'm also going to pause here for a second and just remind you all that even though this is review material, it is a good idea to try to include it in your notes. So I know I'm moving kind of quickly, but if you want to try to draw some basic pictures in your notes or create a mind map, a graphic organizer of sorts in your notes with this information, that's a great idea. And make sure you leave some space in between them for reasons that will become obvious in a few short minutes. All right, so at the end, maybe you'll take a screenshot once we finish this slide and you'll be able to go in and draw it, complete your drawing later, or you might want to draw it as you go. Totally a matter of preference, but I do think this is important information for you all to be able to have. Okay, the chloroplast. What's the function of the chloroplast?
It makes photosynthesis.
Yeah, it does photosynthesis. And remember, this is only present in plant cells. We only find chloroplast in plant cells. The vacuole. What does the vacuole do?
Stores water and other nutrients.
Wonderful. Thank you, Carolyn. Nutrients, waste, and water. And as we saw on the last slide, the vacuole inside of a plant cell is typically larger. We're going to talk about the reason why that is coming up soon. All right, what about the cell wall? What's the role of the cell wall?
To keep the plant from
Keep
expanding.
to protect.
Protect. I heard something, Janelle, kind of got cut off there. I couldn't tell exactly what you said, but said keep something. Maintain water pressure is an important function. Protection, support, all of these things. But it's in general a rigid outer layer that we would only find in plant cells as well as some prokaryotic cells. The cell membrane, what's its function?
It controls what goes in and out of the cell.
Wonderful. Yeah, and I'm gonna throw in another word that's a synonym of controls here. I'm gonna say regulates, because regulates is one of our Sternger words. But I appreciate that, Abigail. And we're gonna talk a lot more about the cell membrane in our next unit. It really has a very important function. And then I think, okay, that's it. So now we want to focus on some of the relationships, because that standard tells us we can't just know the structure, we can't just know the function, but we also need to know how these organelles depend on one another. What is their relationship to one another? So let's take a look at this. Can anyone think of the relationship between the nucleus and the ribosomes?
The nucleus has the DNA, and the DNA is like the instructions to make the proteins.
Wonderful, wonderful, Janelle, excellent. Yeah, so the DNA is the instructions to make proteins. I always think of my analogy of the cookbook. You can come up with your own analogy, whatever works for you, but the DNA is the instruction manual that will eventually lead to the protein. All right, we're going to talk about this. This looks pretty simple. This flow chart here, DNA to RNA to amino acids to proteins. Probably our most important unit of the class is dedicated to this idea. We'll talk more about that later. Okay, what's the relationship between ribosomes and the cell membrane? Kind of push yourselves to think about that.
Doesn't the cell membrane kind of like transport the ribosomes around the cell?
Wonderful, yeah. So, and not just around the cell, but also outside of the cell. So some proteins are meant to travel from one cell to the next. In order to do that, obviously they're going to have to pass through the cell membrane, and the cell membrane ultimately decides which proteins can leave, when they can leave, and how many of them can leave. So there's a clear relationship there. Okay, both the cell membrane and the cell wall are considered outer layers, and so they both have a role in support. What's the relationship between a vacuole and a cell wall? Again, push yourself to think about this one. I'm going to refer to my plant that I had sitting there. This might help you kind of think about the relationship a little bit. Any ideas about the relationship between the vacuole and the cell wall? So let's think about this. Most animals have either an internal skeleton or an exoskeleton that gives them shape. Plants do not have those bones. There's no skeleton in these leaves. There's no skeleton in the stems that allow them to stand up. So their mechanism of actually maintaining structure and standing erect like this is through water pressure. And that's why they have such large vacuoles. They need water pressure to push the cells basically as tightly as they can go in order to stand up. All right, so the vacuole has to provide additional structure to the cell by maintaining water pressure, and that's why the vacuole is so big. A plant that gets wilted, and you can see I've got some leaves down here that are dying. They've wilted a little bit. Those plants are probably, they weren't getting enough water. these leaves haven't been getting enough water or enough sunlight in order to maintain that water pressure. So they start to droop. You've seen drooping plants before. That's an indication that your plant needs some water. Okay. What about the relationship between vacuoles and chloroplasts?
It provides the water for photosynthesis.
Wonderful, yes. Photosynthesis needs three things to happen, and we'll talk about this in a couple of units. Photosynthesis needs, number one, sunlight. Number two, it needs carbon dioxide, which is a gas that's in the atmosphere. And number three, it needs water. So the chloroplast is storing water not only to maintain water pressure, but also so that the plant can do photosynthesis. Oops. And the end result of photosynthesis is, of course, glucose. What's the relationship between chloroplasts and mitochondria?
Mitochondria give the chloroplast energy.
Okay, yeah, we can say that the mitochondria give the chloroplast energy, which is true, and that's actually like a if you take higher level biology courses, you'll see exactly what happens in that relationship. But going the other way, chloroplasts provide the food to make energy, so you need glucose to make energy. So this is the cool thing about plants; they're kind of self-sustaining. In fact, if you put a plant inside of a large sealed bottle, and you give it some soil and you pour some water in it, it can actually live like that and continue to grow and become its own ecosystem without any additional input of nutrients or air or water. Because what happens is that it's just going to recycle the materials that it needs. So photosynthesis, like I said, requires three things: sunlight, water, and carbon dioxide. The cool thing about it is that the products of photosynthesis are oxygen and glucose. Those products are the same things, the same two things that are needed to make energy. So once we make glucose and oxygen, we use those two things to make energy. And then when we make energy, there are two products: water and carbon dioxide. So it's just a cycle. It uses what it needs, and then it uses whatever it produces again in a cycle. And so it's a really cool relationship. We'll look at that more later in the course, I'm sorry. But chloroplasts provide the glucose to make energy. And then one more: what's the relationship between mitochondria and ribosomes? It's a pretty simple one. Anytime you're making something, you need energy, right? So to make a protein requires energy. So mitochondria provide ribosomes with a specific type of energy called ATP. We'll talk about that later as well. But anytime the cell is making something, it's going to require energy. And it relies on mitochondria to provide that energy. All right. So this is a great picture to have, a great screenshot to have. And as we learn more and more about the cell and about these processes, you might add on to it. But I love flowcharts because they really can be useful study tools, and they help give you a visual representation of how seemingly disparate or disconnected ideas are actually connected. So I'll leave this up for about 10 more seconds. Okay. The second standard from this unit, prokaryotic versus eukaryotic cells. It states that students should compare prokaryotic and eukaryotic cells in terms of their general structures, plasma membrane and genetic material, and degree of complexity. All right, so let's take a look at this. Thanks for joining us, Sabina. I'm changing the attendance now. So what we have here are two buckets. The bucket on the left is for prokaryotic terms. The bucket on the right is for eukaryotic terms. Let's see if we can place these terms and pictures into their correct bucket. So at the top, plants. Are plants eukaryotic or prokaryotic? Plant cells, specifically.
You?
Yeah, plant cells are eukaryotic. There's a corny saying that one of the other teachers taught me. Eukaryotic cells are you and the plants, meaning animal cells and plant cells. Eukaryotic are you and the plants. What about chromosomes? Where would we find chromosomes? In prokaryotic organisms or in eukaryotic organisms? What do we think? What do we think? No, chromosomes are more organized forms of DNA. So I appreciate you jumping in for that one, Abina. We find chromosomes in eukaryotic cells. All right, because eukaryotic cells are a little more complex, they're a little more organized, they're younger. So we find chromosomes in eukaryotic cells. Where would we find, or which one of these has no nucleus? Prokaryotic cells or eukaryotic cells?
Pro.
Pro, good. Bacteria, would that be an example of a prokaryotic organism or a eukaryotic organism?
Prokaryotic.
Prokaryotic, good. Which one of these has circular DNA in the form of a plasmid?
Prokaryotic.
Again, prokaryotic, good. Which one of these is older, meaning it is less evolved?
Prokaryotic.
Again, 4 in a row, prokaryotic. Good. So we know that prokaryotic cells are less evolved because they've existed for a longer period of time. They were the earliest life forms. Eukaryotic cells, I'm sorry, I just gave it away. Mitochondria are found only in eukaryotic cells. Now we've got 3 images at the bottom. Let's see if we can identify them as being prokaryotic or eukaryotic. So this image on the left, what is this an example of?
Eukaryotic.
Eukaryotic.
Eukaryotic. And bonus points if you can tell me what kind of cell this is.
Plant.
Plant. Yeah, this is a plant cell. Good. And we can tell because of this rigid cell wall, and I can also see 2 chloroplasts, and I can also see a really big vacuole in the middle. Okay, what about this cell? Prokaryotic or
Animal.
It
Eukaryotic.
is an animal. So is it you or pro?
Eukaryotic.
You, good. And then lastly, this cell right here,
Prokaryotic.
prokaryotic. And does anybody remember what this tail is called coming out of it?
Flagella, something like that.
Flagellum, good, yeah. So we're going to talk about that in the next standard. But this is, again, another good slide to have. Take a picture of it, take a screenshot, make sure you have it in your notes. I'm going to leave it up for about 10 more seconds.
What's that cell called on the prokaryotic side again?
This is some type of bacterial cell. Good question. Thank you, Lisa. Okay. So just quickly to review, prokaryotic on the left, Eukaryotic on the right. Does it have a nucleus? Prokaryotic? No. Eukaryotic? Yes. And, you know, had to throw in the Obamas. I'm in the midst of reading President Obama's memoirs, 800 pages long, but I'll let you guys know how it is afterwards. How is the genetic material organized, the DNA? In prokaryotic cells, it looks like a plate of spaghetti. It's very unorganized. It's disorganized. It exists in a circular plasmid or what's called a nucleoid. But in eukaryotic cells, it's very organized, like a nice, tightly wound knot. And we would call it a chromosome. And these chromosomes are found inside of the nucleus. Does it have membrane-bound organelles like mitochondria, chloroplasts, and vacuoles? Prokaryotic cells, no, they do not. Eukaryotic cells, yes, they do. Okay, keep going. Prokaryotic cells, are they old? Eukaryotic cells, are they old? Prokaryotic cells are much older than eukaryotic cells. Like Kevin Gates says, they're really, really old. Eukaryotic cells, nah, not very old. Somebody else join us recently? Nico or Sally? No. And then how big is it? And keep in mind, big is really a proxy for how complex the cell is. So the bigger the cell, the more complex it is, the more stuff it has inside of it, the more function it is responsible for. So prokaryotic cells are really, really small. Eukaryotic cells are relatively huge. All right. And then our third standard from this unit was about the cellular adaptations. So this states students should explain how specific cell adaptations help cells survive in particular environments. Focus on unicellular organisms. We've got a column on the left side that is asking for the name of the cellular feature. And then we've got a column on the right that is asking how does that cellular feature help an organism to survive? So the first one we covered it's called an eye spot. How does an eye spot help an organism to survive? What does it do? Wonderful. It responds to light. It detects light. And it also allows for something called phototaxis. What is phototaxis? What does that mean?
Movement.
Response to light.
Movement, specifically movement in response to light. So thanks for tag team and JT and Janelle on that one. The second cellular feature we're looking for allows for food ingestion. What's that called?
Pseudopod.
A pseudopod, good. Pseudo meaning fake or temporary, and pod meaning foot. So a temporary fake foot. Cilia. What do cilia do? They really have two functions.
Environment.
They sense the environment, and what else can they do?
Allow movement.
They allow for movement. All right, so those two things together combine to create an ability that we didn't really talk much about, but it's called chemotaxis. Does anybody know what chemotaxis would be?
Wasn't it a response to chemicals or something like that?
Wonderful. So yeah, chemotaxis, thank you, Janelle, is movement in response to chemicals. So a cell might really prefer to be in a salty environment. They've got to use these cilia to detect the level of salt in their environment and then move towards the saltier environment. Or a cell might really like sulfur, a sulfuric environment. So they've got to detect the amount of sulfur and move towards it. Or a cell might really hate acidic environments. So if it detects that we've got a lot of hydrogen in the environment, then I've got to move away from it because that means it's really acidic. So cilia detect what's going on, they sense it, and then they allow for movement towards something or away from it. What's the cellular feature that allows really only for cell movement? That's its only job.
Flagellum.
Flagella.
Thank you, Abigail. Thank you, JT. It's called a flagellum, singular, flagella, plural. And then lastly, contractile vacuoles. What's their primary job?
A storage sac that can hold water.
They hold water. Did you add something, Janelle?
And pump it out.
And they pump it out. Good. So contractile vacuoles expand to take water in, and they contract to pump water out. That is a process that requires energy. So there's another kind of connection that we can draw between mitochondria and the vacuole. Mitochondria have to produce energy. They provide some energy to the contractile vacuole so that it can pump. Pumping is not a spontaneous reaction, meaning it does require energy. Okay? Keep that in mind. That's something we'll talk about in our next unit as well. Pumping requires energy. So leaving this up for 10 seconds so you guys can grab a picture of it or write it down. Okay. So we want to revisit the objective, which asks us to clarify any and all misconceptions they may have about how cells complete the Sturgeon processes. So let's see how well we can relate these concepts back to our 8 essential life processes. Sturgeon. The S in Sturgeon, of course, stands for synthesis. So share out. Where and how do cells synthesize, aka make, new things? Can someone think of an example of how cells make new things?
The ribosomes.
The ribosomes, that's a great example. Ribosomes are making new proteins. Can anybody think of any other examples?
Chloroplast makes glucose.
Wonderful. Thank you, Carolyn. Chloroplasts make glucose. They make food for plant cells. Any others? Okay. We'll talk more about some other examples as well. We can think about how cells create new DNA. We'll talk about that later. We can talk about how cells create copies of themselves in some cases. Keep those two ideas in mind. The T in Sturgeon stands for transport. So 2 questions here. How do organelles move within cells? So the actual organelles inside of a cell, how do they move around? And then the second question, how do cells move towards and away from one another or from a specific environment? What can you think about? What's the we don't really typically think of it as an organelle, but what's the feature of a cell that allows for movement in the entire cytoplasm? Excellent, Abigail. So that's one that we didn't mention today, but Abigail reminds us of cytoplasm. It's a jelly-like fluid that allows for movement inside of the cell. Okay, so cytoplasm is special because it's not runny like watery, but it's also not as, what we might say, viscous as like peanut butter. It's somewhere in the middle. So things can move around in the cell, but they can't move around so much that they end up damaging themselves or other structures within the cell. So cytoplasm is super important and it provides for internal transport. Can we think of any features that allow for external transport for the actual cell to move around? We just talked about a couple of them.
Transport and passive transport.
Janelle, I heard you say something. What'd you say? We just talked about a couple of them with our unicellular adaptations.
The flagella?
The flagella, yeah. Flagella are a great way for cells to move around from place to place. Cilia also provide that, and some might even say the pseudopod provides that as well, because it allows the cell to move and grow out from itself to capture prey. All right, so transport is a cool one. We've got several examples of that. What about excretion? That's the E in Sternger. How do cells store waste?
Vacuoles and
No, not one.
Inside of a vacuole. Yeah, that's a pretty simple one. Regulation. This was one that I think Janelle gave me the word control, and I said I'm going to use the word regulate instead because it's one of our Sternger words. So what feature of a cell allows for regulation?
The cell membrane.
The cell membrane. Thank you, Melissa. So it allows some things to come in and some things to go out. Even things as simple as heat. It can control that to a certain degree. How do some cells digest food, and then how do some other cells make their own food?
Chloroplast?
Yeah, so some cells use chloroplast to make their own food. What about other types of cells?
ribosome.
Okay, ribosomes can make proteins. Yeah, we can. Go ahead, Josue. What was that?
Mitochondria.
The mitochondria. Yeah, we need mitochondria to make energy as well. We might also think about the pseudopod, right? Really the sole role of the pseudopod is to capture prey, to capture food. And then we don't talk about it much in this class, but there is another organelle called a lysosome. L-Y-S-O. Oops. Hold on. I don't know what just happened. L-Y-S-O-S-O-M-E. A lysosome. A lysosome is an organelle that has a bunch of enzymes inside of it. And those enzymes just go around breaking things down, making it easier for the food to take nutrients from it. All right, so that's a cool organelle to know. Growth and development, we'll talk more about that. That's the G in Sternger. We're going to bring that up when we start talking about reproduction. Again, we'll talk about reproduction more on Unit 5, so keep that in mind. The last R is respiration. You guys have talked about it a lot, but cells use mitochondria to do this process of respiration, to make energy. They use oxygen and they use glucose to make ATP, which is energy for the cell. All right, so we've got 15 minutes left. That should be enough time for us to get through a Nearpod session. I just want to ask some questions of you guys and start to gauge how well you're understanding things. So you can either go to join.nearpod.com and enter the code on your screen, or you can click on the link that I just sent to the chat. So join.nearpod.com. The code is 3P8D7. And let me make sure I'm on the right slide. All right. So as promised, I wanted to revisit the essential question from the day. The essential question states, what are the features, or it asks, what are the features of different types of cells that make life as we know it possible? So we've got 3 people in the Nearpod so far. Oh, no, we've got 9. Sorry, my computer is loading slowly. Excellent job, folks. Thanks for being here. We need 6 more people until we've got everybody on the call in the chat or in the Nearpod. So thanks, Sabina. Thanks, Abigail. Thanks, Araceli. Thanks, Carolyn, Dejan, JT, Janelle, Lisa, Melissa. So I'm waiting on Tyshawn, Sienna. Let's see, and several others. It's going to be hard for me to count everybody. but we need 5 more people to join, and then we're at full strength. As you all are joining, consider this question. I'm going to open up this collaboration board. I want you to type at least 2 different answers on this collaborate board. Give me at least 2 different answers to the question. Give me a feature of cells that make life possible and explain what that feature is or what it does. And then as you start to see your classmates add some of their ideas, you can like the messages that they add as well. DeJohn went ahead and was the first like ribosomes because they create protein. Excellent. Okay, Abigail is jumping in and saying mitochondria because they provide energy. Wonderful. Keep going, folks. What features can we think of? JT, vacuoles because they store water and waste. Wonderful. Chloroplasts, they give plant cells energy. Bonus points if you can start to make some connections between these structures. Okay, let's get some more. I don't know if this maybe my computer is loading. Cell wall because it helps protect and provide structure for the plant cell. Thank you, Carolyn. Is it set up so is it possible for you all to submit more than one? Yeah, JT just did. Good. Mitochondria, Janelle, nucleus because it holds the DNA, and DNA is wonderful. Yeah. Thank you, Janelle. It controls the whole cell. Thank you, Lisa. Oh, my back. Yeah, we got some people jumping on that cell wall. Big fan of cell wall. Vacuole and cell wall for the structure of plants. Good, thank you, John. Vacuole provides water for the chloroplast to carry out photosynthesis. Excellent. We're making some vital connections. Vacuole because it contains the water that chloroplasts need to do photosynthesis. Go ahead and wrap up if you're still typing your thoughts. Let's take 15 more seconds to wrap up and then we'll move on. Good. Cell membrane because it controls what goes in and out. Thank you, Lisa. So we've got some excellent contributions here to this Collaborate board. I want you all to consider all of these and even more based on the notes that we've taken this week and during this review session. All right, now we've got a quick 4-question quiz. You've got 3 minutes to answer the question. It's specifically about standard 1.1.2, which is about prokaryotic versus eukaryotic cells. 3 minutes. Okay, we're about halfway through. I see some of you are finished. Excellent job, Carolyn and Dijon and Janelle. Keep going. Araceli, you're doing great too. Thank you, Josue. You're jumping in with some good answers. Good job, Melissa and Habina. Keep going, folks. A little over a minute left. Okay, 10 seconds left. So wrap up if you're still working. Thanks for joining us, Lance. Excellent job, folks. We did really well here. These were some difficult questions. This is the difficulty that you can really expect to see on the test. So if you didn't perform exactly how you wanted to, don't be too discouraged. At least this is just to, again, get you guys acclimated and familiar with the style of question that we ask on the test. And if you didn't do well, then this might be one of the topics that you focus on this weekend while you study or you do your asynchronous assignments. Prokaryotic cells versus eukaryotic cells. Let me see if we did. Oh, it doesn't show you your score? That's odd. I'm not sure how to, because if I don't know how to show you without showing everybody else. So here's the overall class performance. That should be showing up on your screen now. Thanks for joining us, Tayshawn. All right. And again, just really quickly, prokaryotic cells have no nucleus. Eukaryotic cells do have a nucleus. Eukaryotic cells have a membrane-enclosed nucleus as well as membrane-bound organelles like mitochondria. Both of them have ribosomes. All right, so this is just a really simple image. All you need to do is search eukaryote versus prokaryote on Google, and it'll come up with a lot of good stuff for you to do some comparison on your own. All right, let's do another quiz. This one is about cellular adaptations. Again, 3 minutes, 4 questions. All right, halfway through. Some of you are done already. Good job, Dijon. Excellent job, Lance. Good job, Abigail. Melissa, keep going. Carolyn, keep going. You're doing well. A little over a minute left here. All right, 25 seconds left. Most of you are done. We did really, really well on this one. Those of you who are still working, try to finish up. Again, this is just to get you familiar with the types of questions you'll see on Monday. 5 seconds. Good job, Ashley. Good job, Abina and Araceli. Good job, JT. Okay. So here's how we did here. Excellent job. All right. I know we've only got little under 2 minutes. Here's what we're looking at here. Here are some flagella in a microscope. Here is a contractile vacuole. Flagella help with movement. Contractile vacuoles pump water out. We can see these red eye spots here, which are detecting light for these euglena. Okay, one more quiz. Let's see if we can get it done. I know we don't have much time. Did it start? Oh, I guess I didn't put a time limit. So let's see if you can get these 4 questions done, and then you guys are good to go. Make sure you're working on your asynchronous assignments if you know you still have some of them done, that you have not done, I'm sorry. Good, we jumped off to a strong start with that first question. Excellent job. Okay, so it is 1:20. Like I said, I'd like for you guys to just knock this out, finish it just for the practice. But if you need to go, take your 10 minutes before 4th block. That is understandable. Enjoy your weekend. I appreciate everybody really participating today. Excellent job, Carolyn, on this last quiz. Excellent job, Abigail. Josue, you're doing great. Janelle, you're doing well. Peace out, Lisa. Have a good weekend. Good job, Lance. Good job, Tayshawn.
See you Monday, Mr. Red.
See ya.
I got a test on my birthday.
I know. I'll be sure to celebrate with you in some way.
See you Monday.
See ya. Excellent job on this last one, Sway. See you, Lance. Thanks.